Welcome, everybody. Thank you for joining. I see a lot of familiar names popping in and some new ones too. Really glad to have you with us today. I am so excited that today with our friends from the Rare Epilepsy Network, represented by Christina, we are having a conversation about what the future is for looking at how we can expand access to clinical trials and new treatments across the DEEs. And this is something that is near and dear to my heart as someone who works in Deep Connections and the Inchstone Project. And Christina has had many years of work across rare epilepsy different groups. This is really a conversation. And Randall, okay, thank you for joining us today from Longboard Pharmaceuticals and sharing the story about your work. So maybe you guys can just quickly do quick intros about yourself to frame the conversation. So Christina, we'll start with you.
Sure. Well, thank you for having me. Pleasure to be here. So I am the founder of the Lennox-Gastaut Syndrome Foundation. I founded the organization in late 2007. I have a brother who's 36 years old with Lennox-Gastaut, and currently I'm the chair of the Rare Epilepsy Network. And I also serve as a board member for Coalition to Cure CHD2 and an advisor for Families SCN2A.
Great. Thanks. And Randall, let's hear all about you.
Yeah, hi, everyone. I'll give you the short version, not the one from my mom. So I'm Randall Kaye, the Chief Medical Officer at Longboard Pharmaceuticals. I've been in drug development for over 30 years now, so I have like a super fun, interesting job. My background is I'm a pediatrician and an immunologist by training, and I can't wait to tell you our story.
Well, it is a fantastic one, and I'm so thrilled. And I've had the privilege of getting to work with Longboard for a little while, just kind of hearing about what they're doing and staying in tune with that. But this topic came up because there are a growing number of rare epilepsies that are being discovered, particularly with genetic causes, as access to genetic testing grows and as the landscape expands for what we're able to do with genetic testing, more and more of these disorders are coming to light. So, I mean, Christina, as the chair of the Rare Epilepsy Network, you see this day in and day out about you guys have how many members and how much is it growing?
Yeah, so right now we're standing at over 120 member organizations that are being represented, plus partners, which are a little bit different than members. And the growth has been incredible. I mean, we're onboarding between 4 and 6 new rare epilepsy organizations a month. And for those of you that remember REN in its infancy, we only started with 10 organizations. And that was about 10 years ago. So to see that growth in 10 years is remarkable. But it also shows that these organizations are forming rapidly and forming for a reason. A lot of the reason, as you mentioned, Gabi, is because of genetic diagnoses and communities just wanting to sort of rally around that gene that's the underlying cause of their epilepsy. And so the Rare Epilepsy Network is really the collaborator or the convener to bring together everybody around research and improving outcomes and support and all those great things that you know so well. And so I think that's it for like the Rare Epilepsy Network angle, but also as I mentioned, I work with a number of other organizations, and it's really interesting to see the growth within those individual organizations as well, just as new members sort of join each individual organization as they find, their genetic cause and really want to find a community of support.
Yeah. And I think, this comes up, and this has been something that I've really latched onto as an equity issue, right? So those organizations that are formed, those are where there are families who have access to resources or time or have the ability to form these organizations. But then each of us is trying to advocate and find treatments for our disorders. And the reality is there's not enough industry, there's not enough to go around for all of us to have targeted treatments in the very near future, right? Down the line, I'm really hoping we get there. But for now, that's why it's really, really exciting, this model that you and the team at Longboard really chose to go with, Randall. So I think it would be great if, we're going to hear from you in a second about kind of this model, about how you did this, and hopefully a little bit about why you chose to take this route too, because to be honest, this is a non-traditional route in the epilepsy space. And, the trend has been that some of these bigger organizations or bigger disorder groups like Dravet and LGS and tuberous sclerosis, where there's a huge, need and more information and resources, that's kind of where a lot of the targeting has been. But it's really exciting that Longboard kind of saw this greater need and kind of said, it's worth investing some time and energy and taking a little bit of a risk to say, how do we make this work so that we're not leaving behind a ton of families who are desperately searching for treatments, many of whom have been through all of them, right? We've gone through the list, we've run out of options, and our kids are still not having seizure control, not to mention other benefits that come from treatments that actually help. So Randall, I'm going to hand it over to you. Unless there's anything else, Christina, you want to say before we get started to frame
No,
a
I would just
conversation?
agree that, the novel approach is really exciting because I come from sort of both experience, right? The Lennox-Gastaut hat, where 4 FDA medications were approved as when I was executive director, to now advising these other groups where they're like desperate. They're like, please, please include us. We almost fit the LGS diagnosis, but not quite. So we don't qualify for these clinical trials. And I think it's really, really cool and really unique to see this approach. So that's all I wanted just to add, Gabi, is it's
Yeah.
really interesting to see it from both perspectives.
Yeah, and not to say that there shouldn't be more treatments for Dravet and LGS, because the treatments they have are great, but they are not solving all the problems. So we still need more across the board for everyone. So I'm really excited to have you here with us, Randall, to kind of take us through how you all did this and what you learned. And I'm going to hand it over to you.
All right, great. So the title slide is important because the title is a new wave of hope for patients with DEEs and other refractory epilepsies. Why is it important? When I did my training a few years ago, might have been in the 90s, but that's just between us. I remember my pediatric neurology rotations. They were the most fascinating. We had these very engaged families and really challenged by new onset, new challenges with their seizures. And we would spend, at that time, when you could hospitalize someone for a period of time, we would spend 7 days, 10 days figuring out what they had and why, and do significant amounts of testing. And I found it so academically interesting until we figured out the diagnosis, which I thought was the pinnacle. It's like, great, we now have a name. And this patient has Lennox-Gastaut syndrome. Like, great, what do we do? And it was like, well, we could use valproic acid. I'm like, well, I've heard of that. It sounds like what we use for a lot of other seizure disorders. Like, well, then what? Well, we could increase the dose. And at that time, it was in the 90s. I think there was a lack of hope because there really were very little, if any, treatment options for these families, and literally decades went by. I think finally the science is catching up. But I'm going to share with you, and go to the next slide, I'll share with you a little bit about the approach that we're taking at Longboard, because I think there's still a challenge, even though the science is sort of catching up. So we're Longboard, we're headquartered in San Diego. We just hired a few more people, so now we're at 60. We went public in March 2021, and the only reason I mention that is that just provides access for capital raises to be able to fund our research. We were a spin out of a much larger company that was focused on G-coupled protein receptor research for a number of decades. On the left is just pictures of us. Our CEO likes to surf, so we're called Longboard. I don't. In fact, at the very, very bottom of this slide, I think there's a picture of all of us on the beach. If you look really closely, you won't see me. I'm off on the side, sitting in a chair with lots of sunscreen and a nice little hat. So enough of that stuff. Let's talk about the compound that we're developing on the next slide. Our medication is called bexicaserin. It's designed to be a next generation 5-HT2C receptor that has greater selectivity and specificity. So the precedent compound is great medicine, fenfluramine, Fintepla, but there are challenges with that. And another one that was in development also, called lacoserin, all of which activate a certain receptor subtype. Well, we had a different approach. Our approach is, if you know what receptors you think are contributing to efficacy, have your drug just focus on that one receptor. And if you have other known drugs that interact with other receptors that maybe they're contributing to side effects, or you have no idea if they're contributing or not, why don't we just go the selective approach? And that's the approach that we took, and kind of see it on that top row, is that we know our compound binds to one receptor, and that's it. And then on the bottom right-hand side, we know those receptors are in the brain. Whereas the other receptors for 2A and 2B are located in different areas and probably are not contributing to towards efficacy. But that's not the only part of innovation that we added in here. We saw a problem. So on the next slide, and Christina mentioned this a little bit. She talked about a lot of good treatment options that are now available. But as Gabi mentioned, even with these newer treatment options, there's still an unmet medical need. Our patients, our kids are still having treatment refractory seizures, and that doesn't even get into the other challenges of non-seizure outcomes. But there are 4 of these conditions: Dravet, LGS, tuberous sclerosis, CDKL5 deficiency disorder, that do have FDA-approved treatments. The problem is, with all the treatments that are available, these kids and young adults are still having too many seizures, or they're getting treatment-related side effects. But the biggest problem is, and a little bit on us as a pharma company, we focused on the larger groups. It's easier to design a study for a group that has 5, 10, 15,000 patients in it than it is for one that has a couple of hundred. But what that means is all the other DEs have no approved treatments. And it would be important to have a broad spectrum anti-seizure medication that had efficacy and safety and tolerability and ease of use data. But no one has any data on those. So we're left with clinicians needing to find a way to find other treatment options for patients that don't have standard safety and efficacy. So there's a traditional drug development approach and a slightly non-traditional. That's actually going to be my last slide, but don't go to that one yet, Gabi. So I'm going to go a little technical with y'all, but I know you can handle it because I've spoken to a number of you individually before. So on the next slide, I'm just going to go into the sort of novel approach that we took. And sorry, this is such a busy slide, but I'm unable as a corporate executive to speak without slides. So I have to have a slide, or I talk too much with my hands, but that's a whole other story. So we designed a study that went in right off the bat to study the disease more broadly. Instead of just an LGS or just a Dravet or CDKL5 study, we allowed patients with a wide variety of developmental and epileptic encephalopathies. So yes, we did enroll patients with Dravet. Yes, we did enroll patients with LGS, but we allowed a lot of others to potentially be included in this early proof of concept study. It's called a phase 1b/2a study because of the early stage and nature of it. The predominant focus is understanding safety and tolerability and pharmacokinetics, the way the body handles it. But we also, because we're going to be getting daily diary information entered in, we also have the opportunity of looking and understanding the potential efficacy of bexicaserin and its impact on seizure frequency. The inclusion criteria and exclusion was made it fairly open to a large number of patients and our participants. We started off, after they were eligible, into a randomization and up titration. So this part's a little unusual. We allowed 4 to 1 ratio. This is most studies have a 1-to-1 ratio of drug to placebo. So this was 4 to 1 because it was more proof of concept. And then once they reached their maximum tolerated dose, they went into the maintenance phase for a 60-day period. They stayed on drug, and then at the end they either down titrated or they were given the option of rolling into the open label extension study. As it turned out, every single patient that completed the study opted to go into the open label safety. So that'll give us some additional long-term safety and efficacy information. that we'll be able to look at. I'll just highlight one thing that we think is important is that in our discussions with the FDA, no echocardiograms were required in the study that we call the Pacific study. That's an important feature because some of the other medicines that are on the market do require an echocardiogram to be monitored throughout. But that's the sort of the promise of a selective drug, is only impact in that one receptor that we think contributes to efficacy. So on the next slide, we're going to talk a little bit about our overall safety data. So 43 patients were randomized to bexicaserin, 9 to placebo. And so the safety set is everyone that received medication. Then we call something the full analysis set, which is anyone that received medication and then had data in the titration period and in the maintenance. So those are the evaluable patient data sets. So I'll report what do we see overall for anyone that was reported to exposed to medication, and then what did we see in terms of efficacy. So we'll start with the safety set. The serious adverse events, there were 4. One patient twice, unfortunately, had an ankle fracture. One patient was hospitalized with constipation because there was required a G-tube was placed, and one patient had increased seizures. This was a patient who had some respiratory symptoms causing the hospitalization, and then I think many of you have had this experience. Respiratory symptoms eventually caused some increased seizure activity, but the way we code it, it gets listed as increased seizures. During the titration period, 16% of the treated patients discontinued due to a side effect. During the maintenance period, a very, very low rate, 4.7%. So what that tells us is, as we learn more about the medication, focusing on how we do the titration period is very important to lower that titration period. We're actually, some of the changes that we're making in future studies is it's a concept called start low and go slow. So we'll go a little bit lower on the dose and a little bit slower with the way we do the titration, and let patients get used to the medication before we do the dose titrations up. Most common reason for discontinuation was somnolence, and you can see those numbers on the right. Somnolence was the most common, decreased appetite, constipation, diarrhea. These are very, very typical adverse events that occur in patients that are treated with ASMs or anti-seizure medications. The numbers, these are not head-to-head studies, but the numbers are very comparable to what you typically see. These adverse events generally mild to moderate, and a good number of them decrease in time as patients get used to the medication. So let's go in on the next slide, and we'll talk about efficacy. So the primary efficacy endpoint, the thing we were looking at, is what is the change in the observed countable motor seizures. So we sort of divide seizures into countable and non-countable, which is a very weird word, because if you can see a seizure, you would figure they're countable, but they probably should be countable and difficult to count. Motor seizure would be a better one. But so we looked overall at the rate that we saw during the baseline screening period, and then we compared the overall treatment group. So this looks at almost a 60% reduction in the frequency of seizures that occurred during the titration and maintenance period, with a backdrop of 17%. So that is a mean difference of 42%, a p-value that almost hit statistical significance, just a tad over the 0.05 number. We look at median, which is that sort of number that's right in the middle, and we were really excited to see this data. But this actually doesn't tell the whole story. The next slide tells more of the story. We wanted to understand what happens when you treat some of these individual DEEs. On the left-hand side, this is what we observed in the Dravet population. Just by the way, it worked. There were no patients that were randomized to placebo, so a 74%, pretty striking decrease in their overall seizure burden. And then we look at LGS and DE other, and also a very substantial drop in the seizure burden. One thing that's important to look at is the delta, the mean difference between the two, nearly identical, 33.4 versus 33.3%. Why is this important? Our belief is, as a company, that you can study this disease broadly. If you think about how clinicians treat patients with a variety of DEs, there is some variability in some medications that may be customized to the specific DE, especially the genetic DEs, where you're doing a disease-modifying agent. But these small molecules should work broadly. And that was a key hypothesis that we're testing. In a heterogeneous group like LGS, do they respond similar to frankly, a bucket or a heterogeneous group of patients with a variety of DEs? And those numbers compare quite well. This is going to be an important discussion that we'll have with FDA as we talk through why we think it's reasonable to study these diseases broadly, because we think we can fix a fundamental flaw is you don't really have to go DE by DE. You really can create these broad-based approaches. I'll show you something that's kind of fun. On the next slide, we have this thing. It's called this is called a dot plot. So this shows every individual patient or participant and what their response rates were. So all the way on the left, you get a sense of individually what we saw with Dravet, and then in LGS and DE other, on the left-hand side in blue is on bexicaserin. On the right-hand side is the placebo group, and it's exactly what you'd sort of expect to see. Placebo group kind of vacillated. Some get a little bit better, some get a little bit worse, but it's kind of random, which is what you expect a placebo response to be. Whereas in LGS and DE other, you see some similarities between the two groups. So I'm visual, and so I had this like visualization in my mind of like, can we gamish them together a little bit? That's a medical term, gamish. And because it looks like there's a really significant similarity. So this is what happens on the next slide when you gamish them together. They overlap. It's just like we thought would happen. So if you compare the groups of LGS, the NDE other that were on bexicaserin, they overlap. You see the biggest bunching occurring between that sort of negative 50 and negative 75 area. And then with placebo, it's just like I described before, some above the line, some below the line. It's sort of pretty random. This is going to be important information that we're going to bring in when we talk with the regulatory agencies to give proof point that you can study these diseases more broadly. It's a fairer approach. I don't have the data to share with you, but if you look at the safety information, the adverse event profile, striking similarities on safety between the different groups as well. I'm going to share with you one more data slide. I think it's just one more. There's another way of presenting the data. Some people think this is resonates better with families. So this asks the question, well, how many participants had at least a 50% reduction in their seizure frequency over the treatment period? So that would go in, and a provider could say to a patient and their family, the goal is to your child is having 20 seizures a month. Let's get it down to 10. Let's get that cut in half. So 60% of patients had a 50% reduction in seizure frequency or more treated with bexicaserin versus a third. So that's a pretty sort of striking finding. I don't have some individual data cuts on that, but that's just to give you sort of a sense of that sort of relative comparison. But the typical endpoint that's utilized for the FDA is to look at what I showed you early on, which is the countable motor seizures is a typical thing that one would need to focus on. So that leads us to what's our thinking for this global phase 3 program. We do intend to be meeting with FDA and other regulatory authorities. It's titled an end to phase 2 meeting. It's not very creative because we just finished phase 2, so it's called end of phase 2. But what it really is all about is about the global phase 3 program. It's what's next. And so I talked to you early on. We could do the standard approach. It's reasonable. Start with Dravet, Lennox-Gastaut. We can choose a third indication, and that's how other sponsor companies have approached it. We could run them a little more in parallel and do them all about the same time. It's reasonable. That is the That is the traditional approach, and after that, continue to go broader to other DEs and come up with a fourth and a fifth and a sixth indication. Honestly, I think that fundamental problem is still there, is when you get to more increasingly less frequent DEs, it will be harder and harder to find a study population that's going to fill up an entire study. We think there's a better approach. We think it's an accelerated approach to studying broadly these DEs. We think it's important that starting with, by precedent, a typical study, which is well codified by FDA and other regulatory agencies, is in Dravet. We expect to be running our second study also in parallel. This is something that we'll be talking with regulatory authorities about. It's like, well, why don't we kind of do what I just showed you in the Pacific study? Why don't we have a broad DE study? Why don't we enhance it with a large group that have sort of like an anchor indication, such as Lennox-Gastaut, maybe 50% LGS and 50% DE other, and then create that beautiful dot plot that I showed you earlier and say their responses are similar. This should provide the kind of data that's required to say you've studied it broadly. That should result in a broad label. This approach is not without risk. We don't know what will happen as we, instead of just enrolling 52 patients, what happens when we enroll hundreds and hundreds of patients. Some patients, you saw it when I showed you the dot plot for Dravet. You see a great response in a couple of patients, and you see one participant really was a non-responder. I mean, that's part of drug development, and that's what you expect. I'm sure many of you have had that experience where some medications worked and some don't. But we think it is the absolute only way that we're going to have safety and efficacy data beyond the core 3, 4, or 5 Ds that are currently studied. I had an old slide, and it used to show 25 or 26 D others. So the vast majority of patients living with Ds do not have these core ones. The numbers actually far exceed it. And then I was talking with one of the outside expert opinion leaders in the field, and she'd said, well, you do realize we probably detected over 500 different associated genetic abnormalities. And as we learn more and more information, maybe some of these D others will actually be categorized as a very specific genetic abnormality. It may be gain of function mutation or a loss of function mutation. I just think increasingly, as whole genome sequencing and stuff comes upon us, maybe a large number of patients that are diagnosed with Lennox-Gastaut will have that specific diagnosis that we're looking for and understand what's really the underlying cause or challenge. So this is the approach that we want to advocate for as we talk with FDA. And our essential view on this is we think it's fair, and we think it's more scientifically sound. At the bottom, this just describes what we're sort of thinking overall for the study. Primary endpoint we talked about was reduction in countable motor seizures. And we'll go to a much broader age group from 2 to 65, probably do weight-based dosing instead of a fixed dose for the lower weight or age patients. We expect to run this study globally. Our last study was in the US and Australia, so we expect to do a fairly broad extension out. And then we'll also plan to have an open label extension study so we can ensure that those families who have contributed to this study will get access after the phase 3 portion, the pivotal portion, into definitively being able to receive our drug in a 52-week open label extension study. So I would love to keep talking, but I'm going to give some time for some discussion and dialogue. I'll just say as an aside, we really are grateful for the support that you provide. I love going to patient advocacy meetings because I just find y'all are just so great and open and welcoming and sharing your stories. I can tell you that when you tell us your stories, it motivates us. Gabi knows this because I can like go back and I like, oh, I just learned this. I just talked to this dad, and we got to go faster. We've got to figure this out. So when you see us, whether it's our company at Longboard or others, tell us your story, share it. It really does motivate us, and we bring the stories back and we motivate other folks. And I've just noted our director of patient advocacy, Sophia Cacciatore, and her email in case you want to do any follow-ups with that. So I'm going to stop at this point, Gabi.
Yeah, thanks so much, Randall. I get very excited about this. I mean, I think it's important for so many different reasons, equity, access. I mean, the reality is that we have a lot of kids who are struggling, dying, and restricting access to treatments is, it just feels totally unfair. And so we're so grateful that you guys at Longboard are kind of taking this approach. And we hope this is kind of the wave of the future, right? You guys have created a path and shown how it can work. And I think, seeing what happens in phase 3 is going to be really exciting. One of the questions that came in was around, LGS is in kind of its own way, a basket trial, right? Because LGS develops secondarily to some other disorder, right? Some other usually genetic disorder. So, and you showed that in your plot, it was like pretty clear, right? So, if somebody has an LGS disorder diagnosis plus some other genetic diagnosis, how does that work? Do they end up, yeah, I'll leave it there.
It's an interesting example that you bring up. It didn't happen a lot in our Pacific study, but it did happen a couple of times. A participant was labeled as LGS, and as we were looking at the inclusion-exclusion criteria, we worked with the Epilepsy Study Consortium, and there were a few that they said, well, very, very consistent with LGS, except not so sure the EEG pattern was consistent with LGS. It appears that it's more consistent with the broader category of DE other. And so we ended up still enrolling those participants, but we put them under the categorization of DE other.
Gotcha.
And frankly, I just think the data just proves it probably doesn't really matter what the label is. What matters is meeting the criteria, understanding the triad of developmental and epileptic encephalopathy, meeting that broad criteria, and looking to see if patients respond similarly. And the key point is similarly. It doesn't have to be exactly the same. Similarly from both a safety and efficacy standpoint. So our Pacific study accounted for that, and we're hoping once we have our discussions with regulatory authorities that they agree with this broad style approach to understanding DEs.
Yeah. A question came in about if you were able to decipher between the types of seizures and their improvements. So, are there specific kinds of seizures that this is good at treating?
No, not yet. So the data set, even though it's like, oh, Randall, it's only 52 participants, it is a huge data set that we're still diving into and looking at. And yes, we did capture the overall types of seizures that were observed. And so that's additional data analyses that we'll be conducting. When you put together a study, you put together a proactive statistical analysis plan. So you tend to focus on the primary endpoint and then the 3 cohorts that we described, but there's more information to learn. It'll help us think through a little bit in our global phase 3 program to see if, and to understand that. But a larger data set will answer that question about what specifically some seizures that respond better to bexicaserin than others. And the current data set will help us explore it and make some hypotheses, but I doubt we'll have the definitive answer until we run these 300 patient studies.
Yeah, much broader kind of sample to be really looking at there. Yeah.
Yeah.
Yeah, there's another question about, if there was bexicaserin, is showing anything that's indicating an exploratory marker based on biological pathways. So is there any sense that maybe this is better for channelopathies or, other kinds of, brain abnormalities, or is it kind of all, all
You,
encompassing?
you just sold everyone on the reason why we want to study these diseases broadly. So the Pacific study won't tell us that.
Yeah.
But running a study, you haven't asked the question, so I'll just tell you. People will say, oh, well, you had 19 patients with DEEs. What were they? And we're actually purposely not putting them out there. We thought going in
Yeah.
that we would get clusters. We work closely with certain patient advocacy organizations. We thought, sure, we're going to get 3 to 5 clusters. We got exactly what one would expect when you offer to study a disease broadly. We got onesies and twosies. And part of the reason why we probably won't put the data out there is we don't want someone looking at the data and saying that N of 1, well, that patient did extremely well. That's what I should expect for a result. Or an N of 1 where one subject maybe didn't respond. We don't want to discount that. We do want to study this broadly. And we think in a study like I described, we will have hundreds of patients with a broad variety of DEEs. We can answer that question. It's possible that there will be some patients that aren't responders. Maybe we'll see a difference in a participant with a certain genetic mutation of loss of function versus gain of function. But our intent is to study it broadly and get that answer, or get it as best as we can. But I can tell you, the current approach will never answer that question. So we expect the regulatory authorities to be sort of open to this kind of approach. I'm not sure I entirely answered your question because I can't yet.
Yeah, I think for now, good enough. We'll talk again after phase 3. I think you'll have a better answer then.
Yeah.
I've got one last question that if anybody has, I see one that just came in. How are you thinking about complementary real world data that would support continued clinical development and laying the groundwork for future post-marketing needs? Interesting. You have to think on that one for a minute.
No, I don't. I'll give you the answer, sort of. So our compound as designed is probably not disease modifying. We expect it to work broadly, and we expect it to work similarly to what we saw in our Pacific study in terms of seizure reduction. But also keep in mind, we only studied patients in maintenance period for 2 months, 60 days. And the vast majority of patients were adults. 20% of the population were adolescents. So these are participants that have been living with their DE for a long period of time. So I'm just going to move into non-seizure outcomes and other outcomes that are important. I don't know if our compound will have an impact on non-seizure outcomes, but I can tell you in a 2-month study, the likelihood we would see it is really, really low. It's just not enough time for a pre-existing condition. The premise has always been reduce the seizures, stop the seizures, give the brain a chance to develop by not being cluttered with this constant electric activity that's going on. It's very, very disorganized. There's no other way. You can't have a seizing patient, even the non-countable ones, and expect that they will continue to grow and develop from a cognitive standpoint. So we think actually, and this may answer your question a little bit better. The long-term data from an open label study is the best shot to see what happens when you're exposing patients to a medication for a longer period of time. We can look at outcomes like CGI, global improvement assessed by clinicians and the caregiver. We can look at quality of life. We can look at sleep, in these 2 or 3 month studies. But if we really want to look at other cognitive changes and behavioral changes, I think it's looking at the open label study where we're able to get in and study it a little bit longer. That's where we think there'll be a lot more interest and value. And I think there's a number of other companies, other sponsors that are starting to look at more longer term impacts. And even the work that you guys have done with Instone, it's a lot to ask. Even the small steps that we talk about, it's a lot to ask in a 2-month period. But I think we can start to show that in these longer term outcome studies.
Yeah, and I'm really excited to see where you guys head with that, because that's also on the horizon too, is recognizing that in these trials, we cannot just be talking about seizures. Seizures are important, seizures are critical, but so is the rest of our lives. And our kids and our loved ones have complex existences beyond the seizures, and that's going to be critical too. And any drug that is maybe working to do something even besides seizure control, that should be available if it's helping, improve cognition and awareness and all these other things. So I know you and I are having conversations about that. Inchstone Project is working on that, and really excited to see where that's headed. One last question, which is any timeline, any thoughts of when phase 3 might come to light?
That's the question that bothers me the most, because even my mom called this one out. So we released, so I just gotta tell you the story. So we released this data, it was January 2nd, and my mom told her all about it. She's like, great, so when will the drug get approved? I was like, well, we need to run another study first, a larger confirmatory study.
Yeah.
And she's like, well, when is that going to get started, and how long will that take? And I started talking about, about a year to get it started. And boy, I got seriously? Like, why does it take that long? So we don't have the 12 hours for me to take you through all the steps that we need to go through. But here's the reality. We'll get it started by the end of the year. In drug development, that's quick, unfortunately. It bothers each of us, working at Longboard, that it takes that long, but it does. It's one of the challenges in drug development, that we release top line data and it's just the first little peek at a couple of tables and figures. And then we start compiling data and information. We hope to meet with the FDA with our global phase 3 plan. And we expect to initiate it by the end of this year. That's a promise. And we will do everything that we can to shave off time, because I know from talking with families that every moment matters, and I promise you, we'll do the best that we can to do it as fast, but as safely as we can. We don't want to just jump into it as we're working on formulation and thinking about dosing, not just down to the age of 12, but we want to dose all the way down to 2. So that's going to require a lot of work to ensure that we're doing it correctly, and especially in that younger age population. I saw one note kind of go across. So I'd anticipate the studies running in the US and Australia. In Europe, there are some other regions that we're looking at as well. We just haven't put that out there, but I think most likely the early start will be in the US and Australia, and then followed probably next year, potentially with the European sites. But because we already have great relationships within the US and Australia, makes it a little bit easier to go a little bit faster.
Mhm. Terrific. Well, thank you so much for your time, Randall, and thanks to the whole team at Longboard for what you all are working on. Your willingness to take take an innovative approach here and be thinking about the broader community that are desperately in need of better treatment. So thank you from all of us. I see there's some thanks coming in there as well.
Everyone.
Yeah, yeah. And we'll keep people posted. We'll bring you more as we know more. So
Great.
thank you. Appreciate it, Randall. Take care, everyone. Thanks for joining us today.